Welcome to the talk, everybody. There's a coherent body of mathematics that's used to model evolution within species by considering the random nature of how gene frequencies change over time. And mathematics has contributed many interesting ideas that have had an impact on interpretation and analysis of population genetics data. And there's also a two-way exchange between the two subjects, with interesting mathematics being developed and pursued, which was stimulated from population genetics. So this talk's going to be a non-technical overview of mathematics in population genetics and how the subjects, population genetics and mathematics, develop together. Charles Darwin was the father of evolutionary biology. He was one of the first scientists to really engage with evolution, and he had a concept of a tree of life. This tree of life included many species that could be placed on the tree, and so I guess he had some sort of idea that species developed from other species. He published The Origin of the Species, which had a very natural selection view that the fittest individuals always survived. So natural selection postulated that an adaptation survived if the adaptation was the fittest. And he had an enormous influence in evolutionary thinking. And this view of selection persisted for a very, very long time. In fact, selection, of course, does happen, and it's well known, but there's actually a lot of randomness as well in the way that evolution works. And later on, we'll see some people who develop that as well. These are Darwin's finches that he was working on from the Galapagos Island. And he looked at the beak adaptation of finches. And he thought, well, there's probably only one finch with one kind of beak, and all the other finches were descended from that finch. And those with the most adaptive beaks were the ones that survived. And I guess he had some view that you could pass on these adaptations which were fittest. He was a breeder as well, so he knew how these characteristics were passed on if they were good. But he didn't know the real mechanism of how it worked. So he didn't sort of know about genes themselves. He just knew that these characteristics were passed on. This is a quote from Darwin. "Seeing this graduation and diversity of structure in one small, intimately related group of birds, one might really fancy that from an original paucity of birds in this archipelago, one species had been taken and modified for different ends." So that was his view, a very selectionist view of evolution. Mendel was a scientist who worked on pea experiments. He had 30,000 peas that he worked on, and maybe people have heard of him before. If you've done any genetics, you would have heard of Mendelian genetics. And he knew that were factors, he didn't call them genes, I think. There were factors, say, little a and big A in a population, and they somehow controlled the characteristics of these peas. He knew that one gene was passed on from males and one gene was passed on from females, and they occurred in pairs. But he didn't quite exactly know what was going on. He had a good idea of the ratios that you could expect of characteristics, and I think he was quite close to really understanding what was going on. This actually occurred during Darwin's lifetime, but I think Darwin didn't really engage with Mendel's results. Hardy was a very famous mathematician who did lots of interesting mathematics, piles of stuff from Cambridge University. He was a professor at Cambridge University, and a very, very famous one at that. He did number theory and all sorts of interesting pure maths. And one of the things he did was to talk about this Hardy-Weinberg equilibrium. And what it says is that if you've got random mating in a population, and P is the proportion of little a genes and Q is the proportion of big A genes, then the Hardy-Weinberg principle would hold, so that these pairs of genes occur in pairs in individuals, and the probability of finding a pair little a was P squared, little a big A was 2PQ, and big A was Q squared in that way. So there's nothing really in this about selection. It's just random mating and how these genes would occur together in pairs. But it's certainly a mathematical contribution in the early days, and an important one here. So, yeah. This is a picture of blood group gene frequencies A and B, and the frequency of these blood group frequencies of A and B, well, they're really gene frequencies, A and B, varies in the world. And so there's a sort of spatial distribution over the world. And you can think that there's a randomness associated with these gene frequencies. And so often it's not sort of exact frequencies that you get in population genetics when you're dealing with genes. There's a very much a randomness associated with them. And that's a very important feature of population genetics. Cyril Wright was a famous pioneer in population genetics, and here he is on his 90th birthday. I hope we can all work to 90. He certainly produced research right up to his 90th year. He was an amazing person, and he worked. Well, let's see his history. So he was around 1889 to 1998. He died at 99, I think. Okay. He was at the University of Wisconsin. He was in the National Academy of Sciences, and he won several medals here for his work in mathematical genetics. In the early days, people often did lots of science. So there'd be people who did mathematics, they'd do sort of practical genetics and all sorts of things. Now, he was one of these people. He studied guinea pigs and all sorts of things like that. So what was Cyril Wright's mathematics? He was important in talking about the random drift of gene frequencies. So I suppose he was interested in gene frequencies in a spatial way, like those blood genes A and B, but he was also interested in just single populations where the gene frequencies just changed over time. They don't stay constant. They will vary quite a bit in a random way. And so he was very interested in sort of quantifying that result. He worked on diffusion processes, one of the early people who did some work on that. And we'll see later more about diffusion processes. And he had this view of shifting valence, which again was a selectionist view. He had the view that certain sort of characteristics made you very much fitter, and it depends on what your genes you had as to how fit you are. And if you looked over this fitness landscape, you'd see peaks in the fitness landscape where you had very fit individuals who occupied these peaks. And he was a bit concerned about how you got from one peak to another peak. So that's, again, a selectionist view, really. I'm not sure whether it's really a view that's held today, but it was what he thought at that time. Sir Ronald Fisher was a very, very important mathematician, and he was an English statistician, an evolutionary biologist, and a geneticist, a truly amazing mathematician, and maybe some of you have heard of him as well. If you've done any sort of statistics, you've certainly heard of Fisher, a very famous person. Here's 3 pictures of him, from a young Fisher to an older Fisher. He actually ended up in Adelaide in Australia, Fisher. This is some of his history. He was at the Rothamsted Agricultural Station. He published the Genetical Theory of Natural Selection in 1930. He was at London University College, Cambridge University, and as I said, he ended up here in Australia at Adelaide University. So Australia has some good connections actually with mathematical genetics, and he was one of them there. Some of his important mathematics that he did was analysis of variance, which told you how to see whether groups of experiments had the same outcome or different outcomes, very loosely. He was interested in experimental design, the method of maximum likelihood, which is a way of estimating parameters from data, fiducial inference, another way of estimating things, population genetics, which we're interested in, the Wright-Fisher model of evolution, which we're going to come to, and he studied Mendel's results. In fact, he thought Mendel's results were a little too close to what they should have been for comfort. There wasn't enough randomness. And he had this view of the fundamental theorem of natural selection here, which I guess it's a bit technical to describe, but what it says, yeah, a bit to describe. But it was a way of saying, describing how natural selection worked in inheritability from parents to offspring. This is the Wright-Fisher model of evolution, which was studied by Wright and Fisher. And it's a very, very important model of evolution, and I'll try and describe this to you, and if you can, try and keep this in your heads, because it's a fairly simple model, and it also is important in what follows. It's a very important model in genetics. So it looks simple, it's a good model. So models, mathematical models in general, are good if they're simple, and they have some features that really describe data, I think. So that's one of these here. And the way it goes here is that you think of a population of n genes. Here you've got n genes, and n's fixed. And the next generation is formed by choosing n genes at random from the previous generation. So here's a picture here where in the bottom generation here, the current generation, you choose your parents at random from the prior generation. We can't all do that, but in this model, that's what you do. That's one way of saying it. And in this picture, there are really 2 kinds of genes, which we'll denote by colours. So maybe blue is little a and black is big A. So you can see what happens here. If you look at it carefully, so this one just has one, this child is one parent here, single parent, or this parent is one child, if you want to think about it that way. So you can think about it really either that way or that way. You can think about parents giving birth and giving birth to n genes at random, or choosing your parents at random. There's 2 ways to think about it. And here, these individuals have the same parent. It could have been that you had 3 of them have the same parent, but by and large, if n is large, what happens is you usually have this picture where you have just one, or maybe have a single join. And it's very rare that you'd have 3 having the same parent. These are not individuals, so we're not thinking here of pairs of genes. There's no sex in this. There's no male and female in this. It's just a very, very simple model here of these genes, which have sort of been all mixed up, if you like. They've been, the sex is disregarded here, and you're just thinking of how tracing back single genes, which you can do, It hasn't got the complexity of pairs of genes in parents. It's a very simple model. And so some individuals here have no offspring, or there are no, you don't choose them as parents. So in a sense, these are going to be sort of lost as you go further down in time. And eventually, if you kept iterating this kind of procedure, what would happen is that you'd be either all black genes or you'd be all blue genes. It would fix from one of these types. That's what would happen in this population. That's as long as there wasn't any mutation. But sometimes there is mutation between, say, the blue and the black and the blue as well. And that might actually stabilise it so it doesn't hit either black or blue. Okay, so that's a very simple but an important model. And it's a stochastic model. It's a model that occurs in probability theory. What about evolution over a long time period? So evolution really only gets going over a long, long time period. It's a long scale process. And so it's not really any good to look at these boxes over a short period of time because you don't see very much. There's not much change. But over a long period of time, evolution happens and things start to move along. And so suppose there are two genes which are called little a and big A now in a population. And x of t here is going to be the relative frequency of type little a genes at time t. But t is measured in units of n generations, and n is large. And so x of t is between 0 and 1. So it's like looking at the proportion, say, of blue types in a box. Here. But it's in this long time scale. And so all the boxes get squashed up here. And you want to plot then the path of x of t as t gets, as n is large. You want to plot the path of xt as t goes on, time goes on here. And so here's a simulation of what might happen. And this is, as I said, this is looking at the frequency, if you like, in the boxes, which have all been squashed up. And you're looking at a long time period in this. And so what happens here, it's really started at point 4, then it nipped down to point 2, and up and down, it went like this. And so here in this graph, this is a simulation, and x of t is the vertical axis, and time t is the horizontal axis. And you can see what's happened here. It's hit the origin 0, it's hit the axis 0 at 0 when at about sort of three and a bit units here in this drift. It could have actually hit the top one here. And so if A refers to blue, then this would mean all the blue ones were lost. And if you actually hit the other axis, all the black ones are lost, in fact. Okay, so the probability of it, actually the probability of it hitting 1, if you started at point 4, is 0.4. But this one hasn't hit 1 at all, it's hit 0. So x of t like this is called a diffusion process, and this is the kind of thing that mathematicians study. They're interested in functions like this. And so you can think of x of t as a random function. It's not just a single random quantity, it's a whole function that's random. And this is one simulation of it. And diffusion processes are very important in probability theory, and they occur in many branches of science, particularly physics as well. Certainly in population genetics they do. You might have heard of Brownian motion, and that's another diffusion process. It's different from this, but it's a diffusion process too. here's some more simulations in a neutral model and what might happen here. So this is just sort of directly from that Wright Fisher model where the boxes are squashed up. And here we go. This one hit 0, this hit 0. They don't go much past 3.5 here, so the expected time to hit is around about here. And then this one actually hit 1, this one hit 1, and it's all of these start from 0.4. Now, you might look at these functions and say, oh, they look a bit strange. Diffusion processes have paths like this which are always very jagged. They're continuous functions, so you don't sort of take your pen off the page here, you keep drawing the function that's going to hit here. But for those that can differentiate, these paths are non-differentiable. You cannot differentiate them. So that's interesting. And they're sort of non-differentiable because you've sort of squashed things up and got this jagged look about it. Here's some more plots from simulations which have what's called balancing selection. In balancing selection, the frequencies are sort of evened out, and so if the diffusion process gets close to the boundary, it gets pushed down again. So if you go up to 1, you get pushed down, or if you go down to 0, you get pushed up again. And the way these go, you still actually eventually hit 0 or 1, but there's a sort of stability about it before you do it. And the time scale here goes from 0 to 30 in these diagrams. It went from 0 to 3.5 in the other. So it can be a long time before you hit 0 or 1, or it still could be a very short time here, about 3.5 again. You could hit 0 or 1 immediately, but you might go for a long while. And that's an interesting, they're interesting plots. Moto Kimura was a very important person who thought of the neutral evolution theory in 1968. And his view was quite different from the selectionist view. It wasn't totally different. He believed in selection as well, but what happened about that time is that a lot of variation was found when people looked at genes. So at a particular locus where some gene was, they might find quite a lot of variance, and more were being discovered. And so he said, well, it doesn't make sense that all these things are supported strictly by selection, because if it was just by selection, it'd be driven towards one particular kind that was most fittest. So he said, well, there's a lot of neutral evolution as well, where genes just drift around and maybe have an adaptation which then fixes. But neutrality was important for him, things just simply drifting around. A very famous geneticist who worked in diffusion processes and genetics. He also worked on orchids. He knew about genetics. And here he is with James Crow, Ronald Fisher, and that's Kimura, the young person here. This is Fisher. Some of the things about Kimura: he worked in theoretical population genetics. He worked in the National Institute of Genetics in Mishima, Japan, 1949. He was at the University of Wisconsin, 1954. Honours, the Genetic Society of Japan Prize, the Weldon Prize at Oxford, foreign member of the National Academy of Sciences, USA, Darwin Medal, foreign member of the Royal Society. He was amazing. His mathematics, the neutral theory of evolution in 1968, diffusion processes in population genetics. He worked a lot on that. In fact, he did an amazing amount of mathematical development in diffusion processes. And about the time Kimura was doing diffusion processes in Japan, also they were being developed in the United States as well. Bochner, who was more a math physics kind of person, was working in the United States. And they didn't ever seem to talk to each other, but this development of diffusion processes was quite fast at this particular time. And certainly Kimura contributed very much to that. And he didn't work in Brownian motion, for example, where other people would have been working in Brownian motion. He worked on these particular processes in genetics. So a little different from some of the physics kind of Brownian motion material. He had a theory of molecular clock, that the rate of evolution is constant over time. He had 660 papers and 6 books. He's an amazing guy. Then what happened here? DNA came along, the discovery of DNA. And so that changed things quite a bit. So you could actually look at very fine level then about DNA. And here's James Watson and Francis Crick, who won the Nobel Prize in 1965 for describing the DNA double helix. But that opened up a whole new scene for population genetics, because you could work out what DNA individuals had and think about analysing that. And today there's massive data sets of complete genomes and all sorts of things, DNA databases. So the scene has changed a lot. And I think in the early days there was hardly any data, very small amounts of data. It was actually hard to get any sort of DNA data or things like that. But now it's there's an amazing amount. And so the focus has shifted a little bit. And the questions now are, what is this DNA trying to tell us? Can it tell us about the history of individuals? What can we find out from this DNA? And I think there's many things to be found out in DNA studies now. Of course, there's many great mathematical people and statistical geneticists working on these areas. This is a historical kind of overview that I'm giving. Maleko here is a French mathematician, and here is a book that was from a conference in his honour. But what Maleko did was to look back in time, and that was an important step, I think. So he was interested particularly in spatial populations of gene frequencies and identity by descent of genes. So he looked back in time. So in the earlier times, really people were looking forward in time and saying, well, what might happen if certain things occurred in populations? How would things evolve? How would evolution work forward in time? And certainly Maleko was starting to look back in time, and today people are certainly looking back in time. So that was a bit of a turning point, I think. Warren Owens has worked in theoretical population genetics. He's famous for what's called the Ewan sampling formula, 1972. He's got a book in 2004. It's a picture of Warren. Some of you may know Warren, so Warren has Australian connections. Here is at La Trobe University. He's Australian, but he's in Pennsylvania permanently now. He's at the University of Pennsylvania, then to Monash University, then back to the University of Pennsylvania, and he's there at present. He's a fellow of the Royal Society, Australian Academy of Science. He won Pittman Medal and the Walden Prize Medal. But he certainly was originally Australian. This is something to do with what's called the Urn sampling formula that I'll try and explain to you. So mutation maintains different types of genes in a population. And in the infinitely many alleles model, every mutation produces a new type. So this is an assumption here that you haven't got two types anymore, but the amount of variation that came along uncovered piles of types of genes. And so you might have not just little a and big A, but you've got a potentially infinite number of types in a population. And the assumption was that every mutation would give you a new type. And I suppose the logic was a bit like saying if you've got a DNA sequence and you had a mutation, it would probably produce a new type entirely because the DNA sequences are quite long and the chance you had any sort of back mutation or something like that would be very small. It'd be more likely to produce something new because there are so many positions in DNA sequences where a mutation could occur. But it's not about sequences yet. It's about just this infinitely many sites model. So you might think of it as a sort of macroscopic model, really, thinking about what's happening at a sort of overall level. But you could say that underneath this there would be DNA, but you're only considering sort of really different complete types of DNA rather than the fine level. And so in this model here, the theta is a parameter which controls the rate of mutation here. And the probability distribution of the number of genes of different types in a sample of n is the Ewens sampling formula. And so, for example, if you came along and you had 3 A's, 2 B's, 7 C's and 5 D's in your sample, and you had 17 genes here all told, what's the probability of having that configuration? Well, it's 17 times 16 times 15 down to 1 divided by 3, because there's 3 here, 2, 7 divided by 5 times theta to the fourth on this increasing product of theta plus 1 up to theta plus 17 minus 1. So that's the formula for this particular example, the way it works. And it's a power theta to the fourth because of 1, 2, 3, 4. And the audience you can't sort of understand how that came about, but it's an interesting and well-acclaimed probability distribution. There's an Urn model which gives you the Ewens sampling distribution. Mathematicians often use urns, and probabilists are quite fond of having Urn models here. And in this urn, the colours represent different types of genes. Mathematicians often use colours to represent different types of objects. That's what we're doing here. So the prescription goes in the following way. Start with one white ball of mass theta in the urn. This is a white ball, and it's what's called a mass theta. Select a ball from the urn. If it's white, you return it with a ball of the new colour. If it's not, add a ball of mass 1 of the same colour as the ball drawn. So we keep drawing it. Every time we get a white ball, we put a new one in. We put the same ball back in, but we put a new one in, new colour. Otherwise, we just put in the old colour again, plus a new one, plus one of the same colour. So it's new if it's white, put the same colour back in if it's not. So the number of balls increases as by 1 every time, but that's what happens. Stop when there are n non-white balls here. Then you're only really sort of interested in the non-white balls, but the white ones controlling the mutations here. The probability distribution of the coloured balls is the same as Ewens sampling formula. What does it mean to have a mass theta anyway? Well, what it means is, suppose you had one white ball and one blue ball. Then if you chose a ball from that with one white and one blue, the probability that you choose the white one would be theta on theta plus 1, and the probability you choose the blue ball would be 1 on 1 plus theta. This is a picture of Fred Hoppe with a mathematical urn. This slide's a little technical for those that know some mathematics, and it's a connection with random permutations. So suppose the general formula that you get then for the Urn sampling formula is factorial n. Well, you want to have n1 to nd genes of the different types here. A1 of the n i's are 1, A2 are 2, and so on. So, yeah, that's what it is. And then, so the general formula is factorial n divided by 1 to the A1, 2 to the A2, n to the A. And of course, 1 to the A1's 1, but we put it in just to sort of make it tidy. A1 factorial up to AN, theta to the D, where you've got D different types, on the product of these values here. Just before I go into random permutations, you can see that what happens here is that theta is involved here to the power D. And so this tells us in a technical way that D, it's the number of types in the sample, is what's called sufficient for estimating theta. So all the information that you could possibly get about theta is actually contained in D. So you don't get any more information from the A's. When Warren first said this, I think it was regarded with lots of scepticism, of course, because why would you ignore these frequencies A1 to AN? But that's what happens. So the connection with random permutations here. Suppose theta's 1, then this probably reduces to this form here, because this is just 1 and this is factorial n, which cancels with the factorial n here, which is a probability in mathematics that a random permutation has AK cycles of length k for k is 1 to n here. And here's the idea of a cycle. If you write 1, 2 to 7 here, and you write a permutation underneath, there are 3 cycles because you go from 1 to 3 to 3 to 1 again, back 2 to 5, 5 to 4, then 4 to 2 again, and 6 to 7, 7 to 6. So there's 3 cycles in that permutation. So that was interesting because that stimulated work on cycle lengths and random permutations. And one of the authors of this book is in the audience today, in fact. This is a book by Richard Aradia, Andrew Barber, who's here today, and Simon Tavaro. And I think it's fair to say that this was partly stimulated by work on the Urn sampling formula. So that's certainly mathematics that's come from that Urn sampling formula. That's quite a formula that occurs in a lot of places. Jeff Watterson was a person who did a lot of very, very good mathematics in population genetics. And Jeff Watterson, Warren Ewens and myself were at Monash University at the same time. And Jeff wrote a paper about Kimura's use of diffusion theory in population genetics. And he's talking about a quote from Kimura here, and I think it's too much to read the whole lot, but at the bottom here it says, Kimura said, after the expression survival of the fittest, which epitomises the Darwinian theory of natural selection, I propose survival of the luckiest, a phrase that best characterises my neutral theory. That was interesting. Sam Carlin and Pat Moran were 2 other people involved in population genetics, Sam Carlin from Stanford. Sam did a lot of work in probability theory, but he was interested in mathematical genetics as well. He was one of the people who worked on the algorithm for BLAST, which is a database searching algorithm for genes. Pat Moran was an Australian mathematician in Canberra, is professor in Canberra, and many Australian mathematicians are descendants of Pat Moran. He had a lot of students who had students themselves, and Australian mathematics owes a lot to him for that. He had a Moran model which is quite similar to the Wright-Fisher model and has the same diffusion limit. Measure-valued diffusions came along, which are important in probability theory. I wanted to describe just briefly what a measure-valued diffusion might look like. Xt, Yt and Zt are gene frequencies at time t, with the sum of these frequencies is 1. They're all relative frequencies, so they're summed to 1 here. And a measure-valued diffusion associates random positions with gene frequencies. And so you've got positions here, x, alpha, with probability x of t. Alpha can be random, beta, y of t, gamma, z of t, and so on. So here you've got you're probably, or maybe used to sort of probability distributions which have fixed values. But here they have random values. So the positions can be random and also the probabilities are random here. They're important in modern probability theory. So there's a lot of stimulation. Stuart Ethier did a lot of work on this. People who worked on these, Stuart Ethier from Utah, Tom Kurtz, Alison Etheridge, Stuart's from Utah, Tom from Madison, Alison from Oxford here, worked on measure-valued diffusion processes. There's a book by Stuart Ethier and Tom Kurtz about Markov processes. It's a famous book. So John Kingman produced a sort of breakthrough idea in the coalescent in 1982. Here's Sir John Kingman, a famous mathematician from Oxford. He didn't only do that, he did a lot of other things as well. And he's an amazing person who's done lots of mathematics and contributed much in public service as well. I'll just let you read that, I think. For us, the coalescent was the important idea, and so this is a coalescent tree of 1,000, and this idea of the coalescent let people work backward in time. It caught on in biology very well, and biologists usually used to think forward in time and maybe simulate Wright-Fisher populations, but now you could actually look back in time and see what happened. And a coalescent tree here, this is 1,000 individuals, and so down the bottom is I always draw my trees upside down like this. Down the bottom is the sample values. Here there's 1,000 genes at the bottom, and 2 genes coalesce when they've got the same ancestor at this point in time. The rate of coalescence is 1 per pair of genes, and coalescent trees always look like this. We've got a lot of coalescences at the bottom. In a short time, then you have longer times as you go back up here, because it's harder to find your ancestors as you go back in time. That's the reason. So this kind of a tree, if anybody knows about branching processes, it's different from branching process trees. The height of this kind of tree is always about 2 units. It's not It doesn't go on forever. And there's no question of extinction here. It doesn't go extinct. This would be called the most recent common ancestor of the tree. It's in a diffusion timescale. It's about 2 units. It's so fast, this coalescence is so fast, you could start off with an infinite number of individuals and instantaneously you jump down to a finite number. So you can think, oh, instead of 1,000, I could have had infinity down the bottom, which is sort of a gap in, it's a jump in thinking really, that you could have a tree with an infinite number of leaves at the bottom. You call these leaves at the bottom as a tree. So here's some points about it here. The genealogical tree of N genes can be traced to a common ancestor. They coalesce when there's a common ancestor of 2 lines. The rate of coalescence is 1 for each pair of lines at any given time. So for K lines, the total rate's K choose 2, because there's K choose 2 pairs. It's so fast you can start with an infinite number of leaves, and it gave a way to make deductions from data back in time. If you look at a coalescent tree, mutations on the tree are what makes different types. And so in this infinitely many alleles model, each mutation produces a new type. This is what we talked about in the Urn sampling formula. And so, for example, here, these 2 wouldn't be carried through. They'd be superseded by this red one. You'd have 3 guys here, 4 really, which were red. Here you'd have 1 black. You'd have these ones all purple from this one, and this green, and this one's blue here. So it'd be a sample of 15 genes, which could be composed in this way. The Urn sampling formula would apply to that sample in that way. So the way that these mutations really, technically they occur at rate theta on 2 along the edges of this coalescent tree, if you fix the edges. So the sort of rate of these occurring, you can think, oh, you put them on according to a sort of Poisson process on the edges. You're probably used to Poisson processes, or some people are used to Poisson processes on a line, but this is a Poisson process on the edges of a tree. Then there's another model which looks at a finer level. So this is like a microscopic level of the infinitely many alleles model, but this is the infinitely many sites model. So this is looking at very fine detail in DNA sequences. And here we've got these DNA sequences here which mutation occurs to, and mutation occurs at different sites here. So the assumption is that you always get a mutation which opens up a new site and then it drifts along. And this is a real data set from Hamer's Y tree here. I think he had a total of 15 sequences here. That was 1995, but today it's minuscule, the amount of data. But still, here it shows here that B is here, B. Then this green one here occurs in C, D and E, so it doesn't get wiped out by that one anymore, but it carries through to all the genes underneath, and so does E here. In fact, you can easily see that you can deduce these sequences from the tree, because all you have to do is look at B. So here's B, you put a red dot there. This green dot, you put it in C, D and E. Purple one, you put in these 2, and this one you put in E. So you can see that you can always construct this one if you knew the tree, but actually you don't know the tree. So, but there's actually an algorithm which says, well, it's a theorem really that says you get a unique tree from the DNA sequences. So you can go that way or you can go that way. You can always deduce what the tree was. In fact, if I gave everybody in this room this problem, here's the DNA sequences, draw me a tree like that, you'd all eventually get it. But there are sort of clever algorithms to do it for bigger trees. This is a set of beta-globin data sequences here, and it's from this paper here. This was data from John Clegg's lab in Oxford, and there's 300 and... Okay. Yeah. There's 326 DNA sequences in the sample and 16 types here of... 16 types, haplotypes here of different types of genes. So this is just labelling what the different types are here. The frequency in the sample of A1 was 104. Then there's 1 of this kind, there's 8 of this kind, and so on. It takes a bit of looking to understand this. If you look at the columns here, there's 2 types in each column. So there's only 18 different columns here, but the data really is about 3 kilobases long. It's about 3,000 bases in this data set. So these are the columns where there's 2 types in each column. So there's many columns which go just T, 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 or G, 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 and so on. And they're not included. Only these columns where there's 2 are included, and they're called segregating sites. And they give you a lot of information about what happened back in time. This is a beta-globin tree here, which is deduced from that data. And you might say, wow, how can you get all that information from that data set? And so there is a tree construction, first of all. So you can do the tree construction. And it's not a coalescent tree, it's a gene tree. It's different from a coalescent tree. And it's a tree which can have sort of more, different degrees from 2 in the vertex there of coming down. It can be more things. It just tells you the mutation history and the vertices of the mutations in this tree here. So you can draw essentially a unique tree from that. And then if you put a coalescent model underneath, you can work out what the times were in this tree, the average times, by simulating this tree, say, 1 million times, and working out the average ages of the mutations and the average age to the most recent common ancestor. So that is what's happening in this particular data set, and it's about 700,000 years ago. There's a big variance in what might happen here. It might have been half the size, or it might have been 1/2 the times the size. This is just an average. But it was an interesting way of doing it. I think it's the first study where you look at the distribution of the mutations conditional on the topology of the tree. It's subdivided into different positions in the world, and this is a sort of generalised Asian group here, if you look at it from Mongolia, American Indians and so on. This is a generalised Asian group with this mutation here. That was of interest. And the oldest lineages are African lineages, as usual in this kind of study. This is another data set here talking about mitochondrial DNA, a very famous paper by Can, Stone King and Wilson saying mitochondrial DNA lived in Africa 200,000 years ago. This is not quite the same data as before because beta-globin is a nuclear gene. It occurs in pairs in individuals. Mitochondrial DNA is just passed on through females. So you get a different estimate here for times. It's not really that there was one female living in Africa at 200,000 years ago. For a start, it's only the mitochondrial DNA ancestor And there could have been other females around before that, of course there were, but the other females might not have any descendants in the current population, so they don't appear. This is the Eve of the current population. And the way the tree appears is in a horseshoe, so the outside is the sample and the inside here is going to be the top part of the tree, or the ancestors in the tree. It's not constructed in the same way as the other tree. It's not a unique tree where the other one was. Recombination is a feature in nuclear genes in general. It wasn't in this beta globin set, but it usually is, where you think that really going back forward, if you look at it sort of going back in time, this individual is really made up possibly of two genes here, where these two genes break and recombine, so during reproduction to form that bit and that bit form that particular gene. So that happens with a probability, say, R, or with probability 1 minus R. This doesn't happen. And recombination break is chosen along the sequences according to a probability distribution. So the green parts are non-ancestral to the current population here, and the blue parts are ancestral. That's a very sort of simplified view of recombination, which is quite complicated in real life. But it leads to what's called an ancestral recombination graph, which is a coalescing branching graph. And the idea is that if you look at a position here and you have recombination, then you put the left-hand part here and you put the right-hand part here. So the left-hand one has some ancestral material from me, and the right-hand part has some right-hand ancestral material from me. So if you know where these positions are, where recombination occurred, you've got the complete history by doing this. And these genes do coalesce as you go back in time. But there's a big complexity about this graph. And really what happens in the graph is the edges represent fragments of these guys down the bottom. These guys get split up into fragments in the edges here. So each edge has a complexity, but it's really fragments. And you can tell what the fragments are if you know where the positions are here. So a lovely, quite complicated graph that's occupied many people, actually. This is recombination rate variation along chromosome 12 in a study by Simon Myers and others, particularly Simon Myers, Gil McVean and Peter Donnelly. And there's some stunning science in this. They found that there was a big rate variation along chromosome 12. And so the spikes here tell you about rate variation. The higher the spike, the more likely it is to break. And this is done with statistical means. This is found from data by statistical means, where previously you could only use experimental methods to determine hotspots in genes. This is really some stunning science. It's just from Oxford. So Peter Donnelly's an Australian originally, but I think he's fairly much permanently in Britain now at Oxford. He won the university medal at the University of Queensland, 1980. He was a Rhodes Scholar. I'll let you read all these things, but particularly he was knighted this year for services to the understanding of human genetics and disease. And he won the Genetic Society medal in 2020 for his research, partly in understanding recombination. That's the British Genetic Society. And so what the science was that he did, which is really interesting, is they found these hotspots, and then Simon Myers in particular searched for what the bases were around these hotspots and found that it was this motif here which was associated with hotspots. And chimpanzees don't have the same hotspots. Don't have the same motif for hotspots. And then they went on to look at sort of evolutionary aspects of this. And another paper for recombination showed that African Americans have these hotspots here, and they found their genes which were actually associated with this motive. So a very nice study, really. And this is the people here. The first three are from Oxford, and this is David Reich from the Broad Institute in America. I think there's many good people who are doing statistical genetics around different countries. Even in Melbourne, of course, we have the Walter and Eliza Hospital, who are doing great statistical genetic stuff and bioinformatics. Then there's the genetics group at Melbourne University also who are doing it. I won't mention particular names because there's so many people really involved. Many people throughout the world are working on statistical genetics and trying to understand DNA data, and that's very important. And there are many good mathematicians who are working on population genetics things, and some of the mathematics which came originally from population genetics has its own life. For example, there'd be lots of people in France who work exclusively on diffusion processes and don't really come back to the genetics at all. So it's definitely a two-way action between genetics and mathematics. It's been important, I think. And I'll leave it at that.